Hi everybody and welcome back to Miss Angler's biology class. I am Miss Angler. In today's video we are doing the much anticipated Life Sciences Paper 2 prediction for the 2024 final November exam. And if you haven't watched Paper 1 yet, I suggest you go back and watch that one first, as we're going to dive into the Paper 2 topics today. Now, I want to remind everybody that I haven't seen the final exam paper, so I don't know what is in it, but I want you to use this video as a starting point for studying, a place where you can check and double check that you've covered everything. And you never know, I might just mention something that you haven't studied, that you haven't gone over, and you think, wow, maybe I should go back and go over that because I don't remember learning that, or I haven't put much attention or focus on that. Another way that I also kind of predict what's kind of coming up in these exams is because examiners have a certain way of asking questions. We change examiners every 5 years, and we're currently in a new group of examiners, and there seems to be like a theme or a kind of intention with their questions. And so I'm going to kind of highlight that to you so that you know what they're looking for, the skills, the content that they want. And most importantly, I just want to make sure that everybody gets the mark that they've worked so hard for. Now, before we dive into the actual video itself, if you are looking for that last minute push across the finish line and you're looking for some intense workshopping on life sciences, you should think about watching my Smart Start series. It is available on my website, Miss Angler, but if you want to watch the first 25 minutes of it, you can actually find that here on my YouTube. It is the very first welcome video on my YouTube page. Essentially, that captures the beginning stages of the whole workshop. It's a 5-hour workshop. I know it's a lot, but think of it like it's basically me teaching you everything you've learned in the entire year. And it also is coupled with support questions to determine whether you're going to get 50% or if you're going to get 80%. If you can answer those questions, then that's a clear indicator that you're ready for the exam. Now, going into this prediction video, I want you to have a little pen and paper ready so you can jot down everything that I say. And maybe there's something that you don't remember learning, and now you can go back and just revise that. And so I hope that some of these questions do indeed come up this year to make your life a little bit easier. Now, we're going to start off with the beginning section of the exam, which is going to be the multiple choice section. Now, again, it's very difficult to determine where they're putting their attention every year, but I think this year we're definitely seeing more pull and attention to things about genetics and DNA. So I want you to kind of really make sure that you're comfortable with the ideas around chromosomes, alleles, genes, and the differences between those words. I'm actually going to go back to those words at a later stage in this video and just talk about where I think these might also come up. But I think you need to note that genetics has one of the heaviest mark allocation in paper 2, and so we would expect to see a lot of multiple choice questions around genetics and even DNA because they kind of complement one another. Another thing that we might see coming up in multiple choice is almost like processes and words associated with meiosis. So not necessarily the phases of meiosis, although they have been known to give you a phase of meiosis and then to ask what it is and what's happening in it. But what I'm thinking more along the lines is them asking you questions like, what is random arrangement? What is crossing over? Which phase does it occur? What is chiasma or chiasmata? More about the processes and what's happening in those processes in the multiple choice, even as far as tracking chromosome number. They love to ask those combination multiple choices, where you have to pick more than one option to see if they all go together. And they do that by seeing whether or not you understand how many chromosomes there are present at certain stages of meiosis, because obviously in meiosis 1 they're still double-stranded, but in meiosis 2 they're single-stranded, so they might be focusing on something like that. Last but not least, I do see for some reason biogeography in evolution just seems to be something that's sitting in the back of my head, and I'm going to talk about it again later in another section as well, where I think it might come up in a longer format. But let's not overlook what biogeography is, and how things like common descent or descent with modification and biogeography can kind of go together, how one supports the other. So please don't overlook those when you are studying. Now, getting into the terminology section, let's remember my favorite thing, which is always true. Terminology determines how well you're going to do in the rest of the paper, because if you can't give a word or you don't know how to use it, then you won't be able to do the longer questions. And I just mentioned now biogeography. If biogeography doesn't come up in the multiple choice section, I really do feel like it's going to come up somewhere in section A. They want to know if you understand biogeography, and it's not really a question that we ask very often in terminology, but I think this year it's going to get its time to shine. Speaking of which, things that don't get a lot of time to shine is going to be the terminology reproductive isolation. I think it's one of those sections in evolution that we overlook. And I suggest that you know all the different kinds or the different mechanisms of reproductive isolation as well. I did mention this earlier, but chromosomes are giving a lot of energy like this year. Like I feel like we need to know chromatid, chromosome, centriole, centromere, like all those words that are similar to one another. If we're not really like confirmed and compacted and like we know those things well, it's often a very easy way to lose marks. Then speaking of that, and speaking of meiosis as well, I mentioned earlier perhaps the word random arrangement might pop up again, and I think it's all linked to the fact that I think this particular examiner or this group of examiners really likes to test the length of your knowledge, like the depth of it versus the width of it. And I think they really want to know, do you know the difference between independent assortment and random arrangement, and can you actually tell the difference and therefore give the word in the terminology section? So let's focus in on that. And then the last thing that I'm seeing this year that could be a potential, because it hasn't been asked in a while, is mitochondrial DNA or mtDNA. Just remember, in terminology, you're not allowed to give abbreviations, so you have to write out the whole word, which means you would have to go mitochondrial deoxyribonucleic acid, okay? And what would the reference point be for that? Well, that would be like tracing ancestry through the lineage of the maternal line. That's probably the like wording that they would use. And if this doesn't pop up now in terminology, I do think it might actually pop up later on in question 1. And you'll see now when I mention the hominid fossil section that might be appearing, that this might link well with that. Now moving into everybody's worst section, because examiners love to confuse you here, is the A, B, both or none section. I've seen a lot of confusion over the years between allele and gene, and we don't seem to know the difference between those two things, so I think you need to focus a little more attention on that. The next thing I think that might come up in A, B, both or none is probably fossils, like hominid fossils, something along the lines of the scientists who discovered them or the type of fossil, like is it an Australopithecine or is it a Homo group? I think you need to know, and you need to go into the exam knowing the fossil name, who discovered the fossil, and where the fossil was found. Okay, those are the 3 things we need to know really well, because I think that's what can happen in A, B, both or none, that can come up. Now, last but not least, in our A, B, both or none, I think maybe meiosis might show up here too. You've noticed that throughout the video, I've kind of kept referencing back to meiosis, and I do think it's getting a lot of attention this year. But they might do something like try and confuse you whether certain processes take place in prophase 1 or prophase 2, metaphase 1 or metaphase 2, anaphase 1 or anaphase 2. And the trick there is, during meiosis, there are certain things that only happen in the phase 1 but not in phase 2. But there are some things that happen in both. So you've really got to be firm in knowing, like, for example, crossing over only occurs in prophase 1, not in prophase 2. Homologous pairs line up at the equator in metaphase 1, but not in metaphase 2. So you've got to be like really careful about that terminology and looking for it in the A, B, both or none. Now we get into the slightly longer questions. Think of these as like those early on diagram questions that you get in question 1. And I'm thinking this time round we're going to have an evolution question, but potentially in 2 parts. And so the first part I'm seeing is something around modification with descent, which, if we're not so sure what that is, or we don't know of previous questions, it's essentially where they give like a structure, like an arm or a wing or a foot, and they show how it's changed over time in different organisms. I think one of the more recent ones they gave was like how we went from land mammals with legs all the way down to aquatic mammals like whales. Like, how did a leg become a fin in a whale? And that's like modification with descent. I think we might see something like that, but very simplified way, because it's still early on in the exam. Again, I have to circle back to meiosis. I think maybe this might also be the opportunity for them to give you an arrangement of meiosis phases, and then what you need to do is put those phases in the correct order, potentially draw the missing phase, although you don't really draw very much in question 1. I've made a note of this in a later question. You'll probably end up having to draw something in this exam. It's either going to be a graph or it's going to be a diagram. And so more than likely and most likely it's a phase of meiosis, but we'll circle back to that one later. Then another common question that takes turns, and this time it should be coming up this year because it's its turn to come up, is a DNA profile. And that can be in the format of like paternity, so determining someone's paternity, or a crime scene, or, and this could be the most like left field one that could come up this year, is they're gonna like determine maybe ancestry through mitochondrial DNA, and like you have to look at the mitochondrial markers and then determine like who's maybe the mother, which would be an interesting spin on things, because you can use mitochondrial DNA to determine maternal lineage in families. That would be an interesting spin if they wanted to make something different this year, but at bare minimum, they're going to include some kind of DNA fingerprint question. Now, last but not least, in this early on section, I think that, again, I'm going to mention it, biogeography, fossils, and specifically like hominid fossils, like where they are found. Maybe we might get a little phylogenetic tree with the fossils placed on it. And you have to like know all of the scientists' names, you have to know where they're found, or potentially you might get a map of Africa and the actual fossils are placed on it, and you need to then be able to read off the diagram, give a little bit more information. It'll be very surface level though, there's no explanations, remember, at this stage for this. Now let's get into the much longer questions. This is where you require a lot more detailed knowledge. And I'm going to start off with potentially something that you've probably forgotten to study. And I suggest you go back because I don't want you to get into the exam and not have prepared this, and that's cloning. Okay, please remember that cloning falls under biotechnology and genetic engineering. And so it is a component of genetics, and they haven't asked cloning in a really long time. They've asked stem cells recently, but because of that makes me believe that cloning is going to get its time to shine. Now here's the catch. Cloning can be two different kinds. You can get molecular cloning, which is when you create like insulin with bacteria and you clone the bacteria. Or you can get reproductive cloning, which is, you know, when you like make a whole new organism, like Dolly the sheep. I suggest that you know that process really well, and you need to know the benefits of cloning. Things like you can make a higher yield of organism, it's healthier, so disease resistant. It can also, if it's like a plant, it can produce more fruit maybe. If it's a cow or something like that, it can produce more milk. So I think we should go back and make sure that we know how to explain cloning and its benefits. The next question that I see coming up this year has to be a pedigree diagram question. Now, a pedigree diagram, remember, is like a family lineage, and you have to follow through to find who's who and what combination they have. Now, a particular disease that they haven't asked in a long time is red-green color blindness. Remember that that is a sex-linked disorder and it affects the X chromosome. They've asked hemophilia so much recently, and also blood groups, that I'm starting to think that red-green color blindness really has an opportunity to show up this year. If it doesn't show up in the pedigree, then it'll be like the genetic cross. So it's kind of like either or. If they don't use red-green color blindness for the pedigree diagram, then they might use another unknown disease. And they're allowed to do that, remember. They will tell you just enough about the disease so that they can ask a question about it. But just be prepared for red-green color blindness to come up in this exam. Now, speaking of genetic crosses, I've noticed that there's a new trend with the examining group that they like to ask alternative genetic crosses. Now, what I mean by that is they're not just always asking really simple genetics like complete dominance, like a black guinea pig and a white guinea pig. What they've really favored recently is co-dominance and incomplete dominance. And I think this particular exam is going to have a heavier focus on co-dominance, which remember is where a characteristic or a trait, two traits, kind of neither of them is dominant and they kind of share equal space. So kind of think like a white flower with red spots situation. In previous years, they've done it with frogs. They've done it with any animal that can like present two colors at the same time. So I think you should prepare for that at the very least, and even go further maybe and do incomplete dominance where you get like a new color. I think we must be prepared for that because it seems to be a hot topic. Now let's get into speciation. Now, I haven't said this at this point in the video, but I just want to remind you, these questions can appear in any order in this exam that I'm giving out now. Like, generally speaking, the genetics goes first. So DNA will be first, then genetics, and that's all in question 2. And then question 3 is generally reserved for evolution in present times, and maybe like a little bit of genetics. But that's kind of how they weigh it out. They've got like speciation and natural selection later in the paper. Speaking of speciation, they haven't asked speciation in an application form in a really long time. So what does that mean? It means that it's not going to be good enough to just know speciation from the guideline off by heart. You're probably going to have to apply it to a situation, like apply it to an organism. In previous years, they ask speciation and biogeography together. There's that biogeography again. But they were asking things like lemurs and potos. They were like the different monkeys that you find in Madagascar and how they speciated. So I think be prepared to take your speciation template answer from your exam guideline and apply it by substituting in those keywords into your template answer. Now, speaking of speciation, we cannot ignore natural selection because they go hand in hand. This year, I've noticed a kind of recurring theme, like a rotation in what they ask. And I think this year they're going to focus on natural selection and potentially natural selection and antibiotic resistance. You see, you have to know one of those. And what I mean by one of those, if you look in your guideline, it says you either have to know antibiotic resistance in bacteria, HIV resistance in antiretrovirals, or insect and pest resistance in insects. And if you think about it, we've rotated through all of those if you look through the history, but we haven't done antibiotic resistant in a hot minute. Speaking of what we haven't also done in a hot minute that they could also piggyback on is punctuated equilibrium. What they could potentially do is give you a setting where they say, well, natural selection is occurring and we are doing it with antibiotic resistance, blah, 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 and bacteria. And then a little sub-question, a little lower, could say something like, using Stephen Gould's punctuated equilibrium theory, explain how he would have explained how antibiotic resistance would have occurred. And that's when you have to go like, well, the bacteria remained the same for a long period of time, and then they went through a dramatic change over a short period of time and became resistant. So you've got to be able to apply your theories of selection to any setting they give you. Now moving into a little more evolution that I see coming up, and I mentioned this one earlier, but I wanted to circle back to it because I think it might get its own like little mini question, and that's going to be reproductive isolation. It seems to have been a new favorite question of our examiner, but this time I think they're going to focus on like courtship rituals as a way in which animals stay separate. In other words, how do you keep species separate from one another? One of the ways is courtship rituals, or it might be, if it's not courtship, like pollination. Like, how do we keep pollinators separate from one another? So how do we make sure that plants don't cross-pollinate between species? I think that's definitely a question that might show up this year. Now, what I haven't spoken about just yet is protein synthesis. And I do think there's going to be like two sections. I don't think it's going to be one big question, so I'm going to speak about it all in one big piece, but it potentially will be 2 chunks. So the first chunk, I think, is going to focus on the differences in structures between RNA and DNA, and I think a lot of people still get this question wrong, and that's why they keep asking it, which is, what is the difference in the sugars of the two, DNA and RNA, the structure, so double-stranded, single-stranded, and much more, and then like what do they code for, or potentially where are they found, like one's in the cytoplasm and the other one is in the nucleus and the nucleoplasm, so like stuff like that. I think you just need to make sure you're able to tabulate that. And then my second point is about protein synthesis specifically, and I think this year translation is going to get its time to shine, like a hyper focus on the ribosome, and linked to that could potentially be a mutation question. They love asking this, you know, changing a few of the codons, seeing how that changes the protein. Does it change the protein? Doesn't it change the protein? So just make sure that you're really well versed on the structures of DNA and translation this year. Then we need to move into human evolution, and kind of looking at a trend that I've noticed is they're taking turns on focusing on different parts of human evolution and the skeleton. And what hasn't had a lot of time or show recently has to be structures contributing to bipedalism. So I'm thinking we should see a focus more on like the location of the foramen, so like a question where you can see the foramen at the bottom of the skull, plus the shape of the spine, so the curvature, so the S shape, plus the pelvis. And instead of speaking about like intelligence in terms of how large the cranium was, because that was a question that's been asked a couple of times now in the rewrite and in the main end of year paper last year, I think they're going to like ask you to like order the skulls from like bipedal to not bipedal, and then kind of ask you what physical adaptations do hominids or Homo sapiens have in order to enable successful bipedalism. And if you have to base it off the pictures they give you, you have to know how the shape of the spine does that, how the pelvis does that, and how the fact, or the location, should I say, of where the foramen magnum is. So please just make sure you're able to explain that, and explain it at least for 6 marks. I think it's going to be a little of a chunky paragraph this year. Speaking of bipedalism, and this is why I want to link it, if they ask such a detailed question about bipedalism, the final thing that they can link it to is the out of Africa hypothesis. Do you see where I'm going with this? They want you to show, oh, you understand how we walk on 2 legs and we're bipedal and this is the structures that support that. Okay, well then what did humans do with their legs? They walked out of Africa. Can you actually describe or give evidence of fossils that show bipedalism based off of where they were located in the world and how that supports out of Africa? Please remember to learn that off by heart from your exam guideline. Now, the second last question is the dihybrid question. The dihybrid used to live all the way back in question 1, but very recently the examiner now has put it later in the paper, which is interesting. I mean, it doesn't actually matter where it occurs, because the dihybrid question is never, ever, ever going to be long, okay? They will never ask you to fill out a full dihybrid question, ever, okay? So what I mean is they'll give you like a half filled out one and you have to fill in the pieces, or they're going to ask you, this is the 2 parents, tell us the gametes. Or work backwards, these are the gametes, what are the parents? And the final thing they'll probably ask you is, if this is the parents' phenotypes, give us the possible babies' phenotypes that are not like the parents, meaning you have to take like all the recessive alleles and put those together to see what kind of combination of baby you could make that's unlike their parent. That's as far as it's going to go, and it's never more than like an 8-mark question. It's never that difficult. Now, last but not least, I've left this to the very end because I mentioned it right in the beginning of the video, but you're going to have to draw something. And I just need to remind everybody every single year that you will probably have to draw a graph. and a diagram in one of the two exams. Generally speaking, if you draw a graph in paper 1, you will have to draw a diagram in paper 2, and vice versa. So they swap. So if you drew something in paper 1, like a diagram or a table, then you're probably going to have to draw a graph in the paper 2. So just depending on what you've done, just remember you have to be able to draw a bar graph, a line graph, and a pie chart. Let's not forget the dreaded pie chart. And you may also be asked to draw either a phase of meiosis, specifically in paper 2, or you might be asked to draw the chromosomes, and be very careful that you're drawing the chromosomes correctly shaded in. It's the only time we really allow shading to show that crossing over has occurred and you've swapped alleles and they've gone from one chromosome to the other. So I think that might be one of the final kind of summing up questions, or they might marry meiosis with natural selection and kind of make them sit together, because in our guideline, it does say you need to be able to know how meiosis contributes to evolution and natural selection. So they might marry that question in together with one another. Now, if you like this video, don't forget to give it a thumbs up and make sure you're subscribed, because I post every Tuesday and Thursday. And I am doing live lessons as we lead up to our final exam in the next couple of weeks. So make sure your notifications are on, because when I go live, you'll be notified, and I always give you enough time in advance to make sure that you can set aside time. Just remember, everybody, I want you to go now and look over those topics, see if there's something that you've missed or that you should be focusing on more. And I will see you all again soon. Good luck with your exams. Bye.